The overall goal of the following experiment is to induce Neo-Islets in the liver of diabetic mice by helper-dependent adenoviral vector-mediated gene transfer. This is achieved by generating helper-dependent adenoviral vectors carrying the islet transcription factor neurogene 3 and the islet growth factor betacellulin. Next, to induce periportal neo-islet formation in the liver, the vectors are injected into diabetic mice. The treated diabetic mice are then tested for normalization of blood glucose, plasma insulin, and the presence of insulin-producing beta cells in the liver. Results from these assays demonstrate that hyperglycemia is reversed in vector-treated mice as a result of neo-islet formation in the periportal regions of the liver.
The advantage of helper-dependent adenoviral vector is a broad tropism and a high cloning capacity. And helper-dependent adenoviral vector does not contain any adenovirus coding region. That's why it lacks chronic toxicity.
The implication of this technique extends towards therapy of diabetes because induction of neo-islets is an effective alternative to islet transplantation, which is limited by donor shortage, graft failure, and alloimmunity.
Generally, individuals new to this method will struggle because transfection process is difficult due to large size of vector plasmid and to minimize the herpesvirus contamination. The amplification and purification process has to be handled carefully.